The question of the week, was it just yesterday I posted that? It was like a week ago. Got my 5 answers. I think the 5th one rolled in about an hour ago. The huge statistical problem with the article that says these 10, was it 10 states? These 10 states had 51% of the school shootings in the country. Is yours one of them? First of all, obviously sensationalistic, but the statistical problem is what? Those are the 10 most populous states in the country. So, of course, they had more than half. Actually, maybe they, statistically, you might think they would have more than 51%, although I guess it's just 10 states. But yeah, so it doesn't account for the population of those states, which is more than the rest of the country. Good, so let's talk about, guess it wouldn't hurt to quickly review. I won't worry about the cell theory, but the cell membrane, because there was some biochem in there, right? The polarity of the head of a phospholipid, right? It's polar because the electrons are held more tightly by that oxygen. And we know that being polar on Earth means that you attract the most important molecule to life on Earth, probably, at least the most important non-organic molecule. So we call that hydrophilic. And then the fatty acid tails, which are very non-polar, and which you already know that water and oils don't mix, and oil is a lipid. Well, this is a lipid too. So those tails repel water. And so what happens, and I'll go ahead and show you this simulation, when you shake a bunch of these up in water, is they start to do weird stuff. Their tails start to stick together because that's the best way they can avoid touching as much water as possible. And so they start to form little clusters like that. And then the clusters will eventually drift together. And they'll start to form sort of a sheet. And what you end up with is this double layer of phospholipids. Double layer of phospholipids that then, because we still have this end and this end with some of that lipid material, some of that non-polar material touching water, we end up getting a circle, really ultimately a sphere. And we call that a lipid bilayer that's making up that. This is a more three-dimensional version. And so that's the foundation of the cell membrane, right? That's the stuff that makes it structurally the way it is. But it turns out there's a whole bunch more to it than that. And so, don't draw this. But it's more like that. Here's the phospholipids, the little gray guys with the little tails. Okay, but then there's a whole bunch of other stuff. And so our understanding of the cell membrane, a lot of times when we start to learn more about something and understand it in a new way, we call it a model of something. And so this is called the fluid mosaic model of the cell membrane. That part you should write. Do you remember the first time you noticed when you were blowing bubbles? The first time you noticed this, that it's like all swirling around. It's not a solid thing, right? This is a liquid, and yet it's holding its shape like a solid. But it's only because it's a really cohesive material And really, all those molecules are flowing around the whole time. You get it? Because what those molecules are in a bottle of bubbles, they honestly look a lot like little one-tailed phospholipids. Very similar, related molecule. And that's how the cell membrane is too. Now there's some anchor points with the cytoskeleton hanging onto these proteins, but this part, the phospholipid part, is constantly flowing around like that bubble I just held up for you. Very similar. Okay, it is not a solid structure. That's the fluid part of the fluid mosaic model. The mosaic part is that there's a whole bunch of other junk floating in there besides just the phospholipids. And that stuff floating in there has functions that we're going to talk about here in a second. The first thing besides phospholipids that's floating in there is actually something that helps the phospholipids stay fluid. It's called cholesterol. Cholesterol kind of gets a bad rap nutritionally because certain kinds of cholesterol can lead to clogging of arteries, but good cholesterol is absolutely necessary for your cell membranes to function properly. But the main thing to talk about in terms of what's floating in there, the main job-doing part of the cell membrane are the different types of proteins that are floating in there. And those are the purple things on that diagram. The cholesterol are the little yellow things with the little tails on them. The big purple things are the proteins. And there's three different kinds I want you to know. And they're going to come back again and again this year. They really turn out to be pretty important to the way your cells function. The only one you can really recognize in this picture is what we call marker proteins. They're the ones with the green things hanging off of them. And it's labeled, the green thing there is labeled as a carbohydrate. And you can see, if you look close, that those are little hexagons. Guess what those little hexagons are? Anybody want to guess? That's your glucose. Yep. That is a polysaccharide chain hanging off of those things. Okay, so the three kinds are very well simply illustrated here. You don't have to draw this, but you might want to, not the membrane part, but the protein part, just simple versions of these might be a good idea. The first one listed is actually the one furthest right, and that is a transport protein. There are several different kinds of transport proteins. This is maybe the simplest one, called a channel protein. It's really just a hole. There's a hole in the cell membrane, and that hole is very specific, usually. Some things don't need a hole, like oxygen, carbon dioxide, and water. They can all get through the cell membrane without any special tunnel. But some things like glucose isn't going to make it across the cell membrane without help. And you absolutely need glucose to live. And so there are glucose channel proteins that let it into the cell. And those look sort of like that. That's transport protein. There's other kinds. There's pumps that pump certain things in and out. There's gated channels that only open under certain circumstances. But the simplest is really just kind of like a protein with a tube through it. Now, the ones with the carbohydrate chains hanging off of them are typically marker proteins. They carry markers that are kind of like cellular identification tags. Why would a cell need that? Why would your cells need identification? Say that louder. So that blood cells don't destroy it. Why would blood cells destroy it? Yeah, because you have a system of specific blood cells, white blood cells. It's your immune system, right? And they are constantly like patrolling your body. There's these killer commando cells that can like poke holes in bad guys, but they got to be able to recognize the bad guys, and they got to be able to recognize you. And so your cells carry tags like this so that the immune system will ignore them. By the way, autoimmune diseases like lupus and rheumatoid arthritis are ones where that system malfunctions, right? Where your white blood cells aren't recognizing the self tag and they're attacking certain cells in your body. Also, organ transplants, this becomes a problem, right? You get an organ transplant, like a cornea is the most common, or something as big and complicated as a kidney or a liver or a lung. You've got somebody else's cells in you now, and those cells don't have the right marker proteins. So what do? What do organ transplant recipients almost always have to do to deal with this? Helps, but in the case of a kidney, it's usually not quite enough. There's two different questions here. The question you're answering is, what do we like to be true of the organs like kidneys that you do get? It's not the antibiotics. I mean, I'm sure they do also take antibiotics, but the drugs that they take are actually immune suppressing drugs. They're drugs that actually keep your immune system from working, because your immune system would absolutely attack those foreign cells because they don't have the right marker proteins. Question she was answering, what do we like to be true of complicated organs that are getting donated? Well, think kidney. You probably know this already. If you need a kidney transplant, who do you look to? Who do you ask? Your family. Yeah, relatives. Because relatives, it turns out marker proteins are genetic. And your relatives are more likely to have some of the same marker proteins as you, so the rejection mechanism won't be as severe. All that because of marker proteins. Then the third one, which is kind of the most multi-purpose, it's hard to come up with a single example, but they're called receptor proteins. Receptor proteins have a little notch in them, a little nook, little spot there, dent-shaped thing. That's a protein with a specifically shaped opening. What does that remind you of? Yeah, it reminds you of the active site of an enzyme, and it should, because it's a similar thing. Except instead of catalyzing a chemical reaction, what happens when the right shaped molecule fits in that dent really depends on which receptor protein we're talking about, but some sort of change gets triggered. When the right thing fits in there, something goes click and causes some sort of chain of events. The best, simplest example I can give you is your sense of smell. And I'm about to tell you something you can't unknow once you know it. But when you smell something, are you aware that it's not some invisible, like, feeling you're getting? That what you're actually doing is taking pieces of the thing you're smelling into your nose? Okay. So what's happening then is that you've got this wide variety of receptor proteins in your olfactory areas that certain smelly molecules will plug into. They're the right shape to plug into the right kind of receptor. And depending on which receptors get plugged into, they fire off nerve signals to your brain that say, hey, that smells like roses, or hey, that smells like poop. And everybody's receptors, it's genetics, so everybody's receptors are a little bit different. And so that's why we experience smells a little bit different from each other. But there are definite types of receptors and combinations of firing of nerve impulses that communicate certain smells to us. So that's one use of receptor proteins. There are hundreds, thousands. Those three, transport, marker, and receptor, you really need to know. You need to know what they do. They will come back again and again. So learn them now, and things will be better later. Questions? Ask them if you got them. Okay, from the very outside of the cell, let's go to the very middle of the cell. What do we find in the middle of complex cells like yours? We find the nucleus. And it's kind of a holy, like not H-O-L-Y, but like H-O-L-E-Y, a holy structure, has holes in it. What What's in there? Like, what's it for? What's the nucleus for?
It's split your DNA into two different pieces.
You say it splits your DNA?
I don't know how to explain it, but like, it's where your DNA is held.
That's enough. Yeah, it's where your DNA is. Absolutely. And you're probably thinking of like mitosis when the DNA gets pulled apart, and that is true. The nucleus is actually going to have to go away for that to happen. But yeah, basically, it's a container for your DNA. And this is where we first bump into the fact that this year I get to teach you something that you didn't know. I might have mentioned it before, but like your teachers have basically talked to you in metaphors about DNA so far. It's the recipe for you, right? Or like if you're a building, DNA is the blueprint. Somebody last period said bricks. No, no, blueprint is good, like that. And fine, okay, but like how? The recipe is the thing you use to bake the cake, right? The blueprint is the thing you use to build the building. How does DNA get you built and maintained and alive? That's the question, and you don't know that. And the real answer, like the short story is like seems simpler than it should be, but basically the short story is the DNA makes you by coding for proteins. That's it. DNA is a code for protein. Doesn't seem like enough. Turns out proteins are so freaking versatile. They're such workhorse, multi-purpose molecules. Not that a single protein does multiple jobs, but that there's a protein for almost every job. That controlling which ones get made when really is enough to basically do the whole deal. Okay. More on that in third quarter when we learn much more about DNA. For now, so let's keep going with the building analogy. Oh, no, okay, hang on, sorry, mixed up. DNA is normally when it's doing that job of controlling protein production, it's kind of unwound, kind of in a thread-like loose yarn form called chromatin. By the way, a lot of things in biology get named before we really know what they are. Chromatin and chromosomes are named that because we didn't see them in cells because cells are transparent. We didn't see the nucleus in cells until we used a stain. And the DNA is the stuff that attracted the stain and became colored. Chroma means color. That's all that means. Is the colored stuff, you know, the stuff that gets colored. You'd think there'd be a more impressive name for the stuff that controls you, but there it is. And so that loose yarn form is good for making proteins, for coding for proteins, not so good for reproduction. When you want to actually make sure that two different cells get exactly half of what's there, you need a more organized form. So you pack that chromatin real tightly into these real complicated, tightly wound X shapes called chromosomes. Yep. You happen to have 46 of them. That is not a special number. Other organisms have different numbers. I think a potato has 72. So obviously, it's not about complexity, right? It's not more chromosomes making more complicated. Although potatoes fairly. So the nucleus itself, as a structure, besides the DNA, it turns out it's surrounded by a double membrane, like not just a lipid bilayer, but two lipid bilayers. So technically almost like 4 layers of lipids. And then there's those holes I mentioned initially. All over the place, hundreds of holes called nuclear pores. Why would there be holes? You've got the most important stuff in the cell. protected by this nucleus. You're just gonna have a bunch of holes. Well, let's go back to the building analogy. You've got the blueprints. Let's say it's a huge set of blueprints for this really complicated skyscraper, and you got them in a room. What are they there for? Why do you keep the blueprints? Why would you keep a copy of the blueprints? For what? What would you want to look back at it for? Okay, so like to go build stuff? Repairs? Building onto it? All that stuff. So you got to have doors in the room, right? These are the doors. These nuclear pores are how stuff gets in to see the blueprints. Now, you've got workers that are going to maintain your building, right? Or maybe build onto it. Do you let them take the single copy of the blueprints out of the room? No. What do you let them do? What do those workers do with the blueprints? They copy, not the whole thing. They take like the part of the blueprints that's relevant to what they're building, right? Just a part of it. Maybe they take a little picture of it, or they copy part of it in some other way, and then they take that copy out of the room. That's exactly what's going on here. Does anybody know what the worker is, if we're being analogous to a cell? What's the stuff that comes in and copies parts of the DNA, takes it out of the cell to actually build the proteins? Lysosomes are outside the nucleus, and they stay outside. I'm glad you know that name, though. It's called RNA. It's the other nucleic acid, right? It's real similar chemically. Slight chemical differences of like an oxygen and one different nitrogenous base, but that difference actually makes it a much more versatile molecule. Not only can it carry information, and it can copy part of the DNA onto itself, but it can take that information out and then do the right shape to build stuff. And it goes and it takes that information to a place called the ribosome and builds proteins. And that process is like that's like 2 chapters long. We'll get to that. But that's the basic idea. DNA codes for protein, and RNA takes that code and actually gets the work done. Another thing that was named before we knew anything about it, the confusingly named nucleolus or nucleolus. First, we discovered that cells like yours have a nucleus because we stain it and there's a dark spot there, right? Because the DNA attracts the stain. Then we discover, hey, in that stained part, there's a part in the middle of that that's even darker. It's like the nucleus of the nucleus. So they call it the nucleolus. It's not that important. Like it's not the thing that controls the whole nucleus, like the nucleus controls the cell. It's just a place where the assembly of those ribosomes I mentioned starts. I don't even know if I would bother telling you about it, except that it's one of those parts that shows up on things like state tests. So you should probably have it in there, in your brain. I think we're going to call that good. You've got your guided reading due tomorrow.